Welcome to Let's Learn Plants. I am so thrilled to have you here for today's explainer, where we're going to explore this totally hidden life or death battle happening right beneath our feet. We're talking about a plant's strategic struggle for a really crucial building block, phosphorus. I mean, without it, there's no growth, no development, absolutely no reproduction. But actually getting it, well, as you'll see, that is one fascinating challenge. Okay, let's just dive right into this. Here is our roadmap for today. We're covering 1, the phosphorus problem, 2, phosphate starvation responses, 3, internal adaptations, 4, meet the transporters, and finally, 5, regulation and signaling. Let's get to it. Section 1, the phosphorus problem. It's totally crucial but stubbornly locked away. So phosphorus is a non-negotiable requirement for life, right? It's literally the P in ATP, the energy currency of the cell, and it's a core structural piece of DNA and RNA. But to understand how plants get it, we need to clearly define our target. Plants don't just absorb raw elemental phosphorus. Actually, they absorb a highly specific chemical form called inorganic phosphate, or Pi for short. Specifically, we're talking about the anions H2PO4- or HPO42-. So whenever you hear me say Pi today, just think of it as the specific form of phosphorus the plant can actually drink in from the soil. Now, you might be wondering, if it's in the soil, why is it so hard to get? Well, it's because Pi is incredibly antisocial when it comes to plants, but super clingy to soil minerals. This is what we call the availability challenge. Even if your soil is completely packed with phosphorus, it has a very low mobility. In acidic soils, Pi gets chemically sequestered or basically locked up by iron and aluminum ions. And in alkaline soils, it gets locked down by tricalcium. So it's chemically inaccessible without a real fight. The plant can't just passively absorb it. It has to actively go on the offensive to extract it. Which brings us to Section 2, phosphate starvation responses. Let's see how plants adapt to this crazy scarcity. Because plants can't exactly just uproot and walk over to a better patch of dirt. They literally reshape their own physical architecture to survive. When external phosphate levels drop, these local phosphate starvation responses, or PSRs, trigger the root system to physically transform. The plant basically says, okay, stop putting energy into growing the primary root deep down. Instead, it massively increases the number of lateral roots and root hairs, spreading them out wide in the topsoil. Some plants even form what are called proteoid roots, which are these incredibly dense, bushy clusters of tiny litlets. The whole goal here is to efficiently forage for Pi by drastically enlarging the root soil surface area. But, just having a further physical reach isn't always enough against those super strong mineral bonds we talked about earlier. So, the roots deploy literal chemical warfare. They actively secrete organic acids, nucleases, and phosphatases directly into the surrounding dirt. It's like they're deploying tiny lockpicks to break down insoluble organic matter and release the Pi. And on top of this, plants also send out chemical signals to team up with beneficial soil microbes, like arbuscular mycorrhizal fungi. They're essentially hiring microscopic fungal mercenaries to help them gather phosphorus when times get really tough. How cool is that? All right, Section 3, internal plant adaptations. Let's look at how the plant optimizes what it already has inside. So the plant really relies on a two-pronged attack. We just covered local PSRs, which are all about enhancing the acquisition of external phosphorus from the soil. But there's a whole second system at play, systemic PSRs. These are all about the plant adjusting its internal setup to improve how it utilizes and recycles internal phosphorus. It's basically the difference between going out and foraging for new food versus strictly rationing the food you already have sitting in your pantry. In these systemic strategies, they're absolutely metabolic marvels. The plant will increase its root-to-shoot growth ratio, prioritizing sheer survival over getting taller. It'll also aggressively reallocate Pi from the roots all the way up to the shoots, using a key protein called PHO1 to load up the xylem. But my absolute favorite adaptation, the lipid swap. Think about this. Cell membranes need phospholipids, right? Well, during starvation, the plant will actually break down its own phospholipids just to harvest the phosphorus. It replaces them with galactolipids and sulfolipids instead. It also activates metabolic bypasses to conserve ATP and tightly modulates its vacuolar Pi storage to maintain perfect cellular homeostasis. It's a total internal rewiring. Moving on to Section 4, meet the transporters, the microscopic vehicles moving Pi across cells. Okay, consider this table your ultimate study map for memorizing the PhD, or phosphate transporter family.
There are 5 main types here, and they're categorized by where they actually work inside the cell. PHT1 is your high-affinity frontline worker sitting right on the plasma membrane, actively pulling pi from the soil. Then you have PHT2, 3, and 4. These guys are busy maintaining the balance inside specific organelles, like the chloroplasts, the mitochondria, and the Golgi apparatus. Finally, PHT5 acts kind of like the warehouse manager. It sits right on the vacuole membrane, strictly controlling the influx and storage of pi. Now, the crucial point here is not to forget these other critical players that operate outside the main PHT family. You have PHO1, which we mentioned a minute ago. This is your main cargo loader, mediating pi efflux so it can be loaded into the xylem and shipped up to the shoots. Then there's VPE, which releases pi out of vacuole storage when the cell is really starving. And SPDT, which is another important plasma membrane transporter involved in uptake and translocation. You definitely want to keep these straight.
All right, on to Section 5, regulation and signaling. Let's see how the plant actually controls all those transporters. So, how does the plant manage this massive fleet of transporters? If it makes too many, it totally wastes energy, but make too few, and it starves. This control happens at 2 levels, making the transporters and destroying them. Transcriptionally, central regulators called PHR proteins bind to a specific DNA sequence, the P1BS element, to turn on the genes that make PHT1. But just making them isn't enough. A helper protein called PHF1 has to physically traffic those new PHT1 transporters out of the endoplasmic reticulum and get them firmly onto the membrane. On the flip side, we have the quality control destroyers, PHO2 and NLA. These are degradation proteins that are constantly breaking down PHT1 and PHO2 transporters based on the cell's current phosphorus status. Which leads us to the grand finale of plant genetics, the MIR399 PHO2 axis. Imagine the plant shoots suddenly realize they're starving for phosphorus. They upregulate a non-coding RNA called microRNA 399. This MIR399 acts as an emergency SOS messenger, traveling all the way from the shoots down into the root system. When it arrives, it targets and suppresses PHO2. Yep, the exact same protein we just learned destroys the transporters. By stopping the destroyer, the plant stabilizes its PHT1 and PHO1 transporters. That means more active vehicles stay on the membrane to frantically pump pi into the plant. It's just a remarkably elegant genetic feedback loop. And that brings us to the most important question for our future. How can we use our knowledge of this MIR399 phospho2 axis, alongside all these physical and chemical starvation responses, to engineer better crops? As our global phosphorus fertilizer reserves start to deplete, mastering this kind of plant physiology isn't just some academic exercise. It is absolutely the key to solving global food challenges and helping our crops thrive in nutrient-poor soils. I really hope this explainer sparked your curiosity about the hidden brilliance of plants. Keep studying, keep asking those big questions, and I'll see you next time on Let's Learn Plants.